Okay, so welcome to this video. In this video what we're going to talk about is phospholipase C enzymes. Okay, so let me outline the structure for this video. What we're going to start off with is a discussion of what phospholipase C enzymes are. Now in this video we're specifically going to be discussing phosphoinositide-specific phospholipase C enzymes, which are the main type of phospholipase C enzymes. Generally when people just say phospholipase C enzymes, they mean phosphoinositide-specific phospholipase C enzymes. So we'll discuss what that actually means. We'll discuss what the target is for phosphoinositide-specific phospholipase C enzymes, i.e. we'll discuss phosphatidylInositol 4,5-bisphosphate, but we'll also discuss other targets for the phosphoinositide-specific phospholipase Cs, such as phosphatidylInositol 4-phosphate and just phosphatidylInositol itself. Okay, right. Then what we'll do is we'll discuss the entire family of phosphoinositide-specific phospholipase Cs, and there are 13 different genes that are known to code phosphoinositide-specific phospholipase C enzymes that are split into 6 different families. So we'll look at these 6 different families. Then what we'll talk about is the catalytic domain, which all 13 of these phosphoinositide-specific phospholipase C enzymes share. Okay, and we'll discuss the basic principles of how you activate the catalytic domain. Okay, so we'll discuss the structure of the catalytic domain because it's got a rather interesting structure. It's what's known as a tin barrel-like domain. Okay, and then we'll discuss how you activate it, so recruitment of the phospholipase C enzyme to the underside of the phospholipid bilayer, and then reorienting it so that the autoinhibitory loop is removed from the catalytic active site. Okay, right. After that, what we'll do is go through each of the 6 different families of phosphoinositide-specific phospholipase C enzymes in turn. So we'll start with phospholipase C beta enzymes, the most famous ones. Then we'll go on to phospholipase C gamma. We'll talk about phospholipase C delta after that, phospholipase C epsilon after that, phospholipase C zeta after that, and then phospholipase C eta to finish off with. Okay, right, so that's the structure then for this video. So firstly, let me get over this first little technical thing, which is that when we say phospholipase C enzymes, which for the rest of this video I'll most likely be abbreviating down to PLC, when people say phospholipase C enzymes without clarifying which specific type of phospholipase C enzymes they mean what are really, strictly speaking, called phosphoinositide-specific phospholipase C, which is written in full PI-PLC. Okay, so what this stands for is phosphoinositide-specific phospholipase C. Okay, and throughout this video we're only going to discuss the phosphoinositide-specific phospholipase C. Okay, so if I just say phospholipase C, I mean phosphoinositide-specific phospholipase C. There is also another type of phospholipase C, which is known as phosphatidylcholine-specific phospholipase C. Okay, which cuts the substrate molecule that the same place as it cuts the phosphoinositide-specific phospholipase Cs cut their substrates, but the substrate is different basically. In this case it's phosphoinositides. In the case of phosphatidylcholine-specific phospholipase C, it's phosphatidylcholine, so I'll just write out phosphatidylcholine here. So the PC there is for phosphatidyl and then choline. So this is phosphatidylcholine-specific phospholipase C, and we're not going to discuss the phosphatidylcholine-specific phospholipase Cs at all in this video. We're going to focus completely on the phosphoinositide-specific phospholipase Cs, which are the more famous ones. Okay, and these are really important in many signaling pathways. Okay, right, so that's that technicality over and done with. So throughout this video, if I refer to phospholipase C, I mean phosphoinositide-specific phospholipase Cs, the main type of phospholipase Cs. Okay, so let's now talk about what all of these enzymes do. Before we actually look at the enzymes, let's discuss what they do, which is that they break down phosphoinositides, basically. They have a specific preference for the phosphoinositide, phosphatidylinositol 4,5-bisphosphate, but there are other phosphoinositides that they can also break down. So I suppose the first thing we need to discuss is what actually is a phosphoinositide. So basically a phosphoinositide is a molecule that is based on phosphatidylinositol. So let's firstly discuss what phosphatidylinositol actually is. Okay, right. So in order to understand what phosphatidylinositol is, we firstly need to know what phosphatidic acid is, okay, because the phosphatidyl prefix means that this molecule is going to contain phosphatidic acid within its structure. Okay, now phosphatidic acid is basically the core for a huge number of phospholipids that are in the phospholipid bilayer. Okay, so in fact, all of the phosphoinositides are phospholipids, and they're going to be present within the lipid bilayer. So here is the lipid bilayer, let's say it's the cell membrane. Okay, so we'll have this representing the outer leaflet of the cell membrane, and this line representing the inner leaflet of the cell membrane. Okay, right. So I'll draw my phosphatidic acid molecule now implanted into the phospholipid bilayer, and it's an important component of the phospholipid bilayer. Okay, right, so let's have a look at what the structure of phosphatidic acid is. Okay, so we won't draw this out in actual chemical structure, we'll just draw a cartoon of it. Okay, so this is my cartoon for phosphatidic acid. Okay, so let me show you the different portions here. So this horizontal line, firstly, in green now. Okay, this represents a glycerol molecule. Okay, now the proper chemist name for glycerol now is what's called propane 1,2,3-triol. Okay, so this is the proper name for glycerol. Okay, and although it's more of a mouthful than glycerol, it's a more useful name because it tells us exactly what we're actually dealing with here, what the chemical structure of this molecule is. It tells us that we're dealing with a 3-carbon molecule, that's the propane, and we've got alcohol groups coming off the first, the second, and the third carbon of that 3-carbon molecule. Okay, so propane 1,2,3-triol. Right, now, these two vertical lines, which are now coloured in orange, those represent long-chain carboxylic acids, or what a biochemist would call the fatty acid molecule. Okay, so these are just carboxylic acid molecules that have really long hydrophobic tails, and these are the lipid components of this molecule. These are the parts which will be extremely hydrophobic and therefore will implant into the hydrophobic core of the phospholipid bilayer. Okay, and at the tip of the fatty acid, you'll have the carboxylic acid group, and this will be bound to alcohol groups of the glycerol molecule by ester links. So this fatty acid molecule here will be bound to the alcohol group of carbon number 1 by an ester link. This carboxylic acid here will be bound to the alcohol group that comes off carbon 2 of the glycerol molecule. Okay, and you've got 2 fatty acids overall bound to that glycerol molecule to make phosphatidic acid. Now the final thing that we need to add on here is the actual phosphate group, which will be bound on to the alcohol group that comes off the 3rd carbon. So this purple blob here, this represents the phosphate group of our phosphatidic acid. Okay, right, so that now is the structure of phosphatidic acid. It's just a glycerol molecule here with 2 long-chain carboxylic acids attached to the alcohol groups that come off the 1st and 2nd carbons of this glycerol molecule by ester links, and then off the alcohol group that comes off the 3rd carbon, you've attached a phosphate group via a phosphoester link. Okay, so that's the structure of phosphatidic acid. Now clearly there is not just one phosphatidic acid molecule, because I have not told you what the long-chain carboxylic acid molecule should be. Okay, so you can actually have them as many different molecules, which means that there is a great number of different phosphatidic acid molecules, but it doesn't hugely affect the properties of the phosphatidic acid molecule, which long-chain carboxylic acids you specifically have in there. As long as they're hydrophobic, that's the key chemical property that they have. Okay, right. Now, phosphatidic acid molecules are a phospholipid. In order to classify as a phospholipid, all you have to have is a phosphate group and a lipid group. Okay, so we've got our phosphate group here, and we've got two lipid groups, which are these two long-chain carboxylic acids. So we certainly meet both of those requirements, basically. So phosphatidic acid is a phospholipid. Okay, right. So phosphatidylainositol, that's phosphatidic acid. So phosphatidylainositol is going to be based on the structure of phosphatidic acid. Whenever you see the prefix phosphatidyl, that means the molecule is going to contain phosphatidic acid. Okay, so we know what this portion means. Now we need to take on the inositol portion. What does inositol mean? Well, inositol, again, is an old name for a molecule that now has a new name. Okay, and the new name for inositol is cyclohexane 1,2,3,4,5,6 hexol. So cyclohexane 1,2,3,4,5,6 and then hexol. And again, although this is a massive, great mouthful of a name, it's a useful name because it tells us exactly what we're dealing with here. It tells us that we're dealing with a 6-carbon molecule where the 6 carbons are arranged in a ring, okay, where all of the bonds between those carbons are single bonds, and then off each one of the carbons in that 6-membered ring, we have an alcohol group, 1, 2, 3, 4, 5, 6 hexol. Okay, and therefore you'll also finally have one hydrogen coming off each of the carbons there. Okay, so that's what inositol is. So I'll draw inositol now, added onto my phosphatidic acid molecule to create phosphatidylainositol. And the way you're going to attach it on is by attaching the alcohol group that comes off the first carbon of the inositol ring to the phosphate group of the phosphatidic acid molecule. So I'll draw inositol like so, as a hexagon which represents the 6 carbons, okay, in the cyclohexane ring, and then I won't draw all the alcohol groups coming off those, but each one of these carbons would have an alcohol group coming off and also a hydrogen coming off. Okay, so there is my picture now of phosphatidylainositol, and it's the first carbon here that is now attached to the phosphate group. Okay, so the first carbon of the inositol ring is attached to that phosphate group via its alcohol group. Okay, right, so that's my cartoon structure of phosphatidylainositol. So phosphoinositides are going to be based on the structure of phosphatidylainositol, basically. Okay, and to turn phosphatidylainositol into an arbitrary phosphoinositide, all you have to do is add more phosphate groups onto the alcohol groups that come off this inositol ring, basically. Okay, in fact, phosphatidylainositol is considered a phosphoinositide, okay, but you can add more structures onto the inositol ring, and that's how you get a bigger category of molecules. So this is, if you like, the core phosphoinositide, but you can add more things onto the alcohol groups that come off the inositol rings, usually phosphate groups, to create a much broader class of phosphoinositides. Okay, so let's have a look at a few examples of phosphoinositides. So we will look at the one that's going to be really important for us. The main target for the phosphoinositide-specific phospholipases, the thing that they absolutely love to break down, is a molecule called phosphatidylainositol 4,5-bisphosphate. Okay, so this is going to contain the phosphatidylainositol molecule, and then all we're going to do is add phosphate groups onto the alcohol group of the 4th and the 5th carbon of the inositol ring to create phosphatidylainositol 4,5-bisphosphate. Okay, now for short, phosphatidylainositol 4,5-bisphosphate is often just abbreviated down to PI for phosphatidylainositol, and I should have mentioned that earlier. That's a common abbreviation for phosphatidylainositol, although of course here we really used it to imply phosphoinositide, so be aware that it can imply both phosphatidylainositol and phosphoinositide, but you're now aware of how similar those two concepts are anyway. Okay, so PI for phosphatidylainositol, and then you put P2 for phosphate, and then you've got 2 of them, so 2. Okay, now, of course, that abbreviation doesn't tell you which alcohol groups the 2 phosphate groups actually come off, so a slightly better, a more rigorous abbreviation is to abbreviate it down to phosphatidylainositol 4,5 bisphosphate, like so. So you put 4, 5 in brackets there, and then you've got the P2, and this tells you where the phosphate groups are actually coming off the inositol ring. Okay, now, however, if you do just see PIP2, you can assume that it means phosphatidylinositol 4,5-bisphosphate. This is the most famous form of phosphatidylinositol with 2 phosphate groups coming off. If you just saw PIP2 without clarification of which alcohol groups the phosphate groups are actually coming off, you would assume that they're coming off 4 and 5. It's the most famous example of this molecule. If someone was talking about another form of PIP2, where, for instance, maybe the phosphate groups were coming off number 2 and number 3, they would absolutely make sure that they put the numbers in the middle, basically, so they would emphasize it. So if you do just see this, assume that it means this. Okay, right, so that's the main substrate of phosphoinositide-specific phospholipase C. So let's have a look at the structure of phosphatidylinositol 4,5-bisphosphate. So all we now need to do is add 2 phosphate groups onto phosphatidylinositol to create phosphatidylinositol 4,5-bisphosphate. So let's do this. So here was the first carbon of the inositol ring, then we just count round 2nd, 3rd, 4th. So we're going to stick a phosphate group off the 4th, 5th, and therefore we're going to stick a phosphate group off the 5th, and that will now create us phosphatidylinositol 4,5-bisphosphate, or PI45P2. Right, okay, so you might be wondering, well, why is that not called phosphatidylinositol 3,4-bisphosphate? Okay, you might be wondering, what is the difference between this carbon here, the 3rd one, and this carbon here, the 5th one? Okay, because on my picture, obviously, there is no difference between the two. However, there is a subtle difference between the 3rd carbon of the inositol ring and the 5th carbon of the inositol ring. So what is this subtle difference? Well, as I say, it's not something that I can get across with this simple cartoon picture, but basically, if you think about the actual structure of this inositol molecule, cyclohexane 1,2,3,4,5,6-hexol, you will realize that there are many different optical isomers of that molecule. Okay, so inositol actually has 9 optical isomers. Okay, and in biology, thankfully, only 1 optical isomer of inositol is actually used. Okay, and that optical isomer that's used is called myoinositol. Okay, now in myoinositol, there is a subtle difference between the 3rd carbon and the 5th carbon. Okay, they are different optically to one another. Okay, but it is a subtle difference. So there is, what you need to take from this is that there is a very good reason for why the 3rd and the 5th carbon are not equivalent, and it has to do with the optical isomerism of the molecule. Okay, now I'm not going to go into that anymore here, but there is a very good reason for why it's phosphatidylinositol 4,5-bisphosphate rather than 3,4-bisphosphate. Phosphatidylinositol 3,4-bisphosphate is a different molecule, a subtly different molecule. Okay, right, so phosphatidylinositol 4,5-bisphosphate then, this is the main target for the phosphoinositide phospholipase Cs, okay, and it's a normal phospholipid that you do have in the cell membrane. Okay, it's not a fantastically abundant phospholipid that you have, not compared to like phosphatidylcholine, but it is a member of the phospholipid bilayer of the cell membrane. Okay, so it is something that you normally have present in the cell membrane at a little concentration, basically. Okay, so now let's discuss what phosphoinositide-specific phospholipase C enzymes are going to do to our phosphatidylinositol 4,5-bisphosphate molecules. Well, basically what they are going to do is they're going to cleave the bond between the alcohol group that comes off the 3rd carbon of the glycerol molecule and the phosphate group. They are going to cut there, basically. Okay, and they are therefore going to create 2 products from this cleavage of phosphatidylinositol 4,5-bisphosphate, and I'll draw these 2 products here. Okay, so one of the products is going to remain in the cell membrane, like so. Okay, and this consists of the glycerol molecule. Here is this horizontal green line, with the 2 long-chain carboxylic acids implanted into the phospholipid bilayer. Okay, now this molecule is what's known as a diacyl glycerol. Okay, or for short, a DAG molecule, a DAG molecule. So D for di, A for acyl, G for glycerol. Okay, and it's called that simply because it has the acyl groups of 2 long-chain carboxylic acids attached to the glycerol molecule, so it is a diacyl glycerol. Okay, so that's one of the products of this cleavage by the phosphoinositide-specific phospholipase C enzyme. The other product that we're going to get is the inositol ring with the free phosphate groups coming off. So let me just show this. Okay, so here is our inositol ring, and here are the free phosphate groups coming off here. Okay, so I'll colour them in again. So here's the inositol ring in blue here, and now we've got a phosphate group coming off the first carbon here, the 4th carbon here, and the 5th carbon here. Okay, right, and this molecule drops into the cytoplasm, okay? This was only attached to the underside of the phospholipid by there because it was attached to the diacyl glycerol molecule previously. Now that it's no longer attached to that, it's just going to fall into the cytoplasm. Okay, and this molecule is what's known as inositol 1,4,5-trisphosphate. Okay, some people will even put the myo in front of there. They'll call it myo-inositol 1,4,5-trisphosphate if you're being extremely rigorous. Okay, so inositol 1,4,5-trisphosphate. We won't go that rigorous though. Okay, and for short, inositol 1,4,5-trisphosphate is abbreviated down to I for inositol, P for phosphate, and then because you've got 3 phosphates coming off, it's IP3. Okay, right, so these are the 2 products of the phosphoinositide-specific phospholipase C enzyme cleavage of phosphatidylinositol 4,5-bisphosphate. You get diacyl glycerol molecules which stay in the cell membrane and IP3 molecules which go into the cytoplasm. Now, these 2 molecules have extremely important signaling roles. Okay, so let me just briefly tell you about the signaling roles of each of these molecules. Okay, so basically IP3 goes into the cytoplasm and then can bind to and activate channels that are in the membrane of an intracellular organelle. Okay, and this intracellular organelle is the endoplasmic reticulum, the ER. Okay, now in the ER membrane you have receptors for IP3 which are calcium channels. Okay, and when IP3 is produced in the cytoplasm, the IP3 can act on these receptors and cause them to open, and that causes the release of calcium from the ER. Okay, so calcium concentration in the ER is much, much higher than it is in the cytoplasm. In the cytoplasm, it's usually ridiculously low at around 100 nanomolar. Okay, so basically, when IP3 is produced, it will bind to the IP3 receptor, okay, and the IP3 receptor is actually made out of 4 separate proteins stuck together, 4 separate subunits, okay, and each one of these subunits has a binding site for IP3. Now, it's not thought that you need 4 IP3 molecules to bind to the IP3 receptor for it to open. It's more around 2 or 3 that you need to bind, but this still isn't fully figured out yet, okay, so you need some IP3 molecules to bind to the IP3 receptors, and then through a gating mechanism that again fully isn't worked out yet, the IP3 receptor then opens in response to the IP3. So we'll simplify it down to just the IP3 binding and then the IP3 receptor opening. It is more complicated than that. The gating mechanism of the IP3 receptor, as I say, it's still controversial, and it's certainly more complicated than just IP3 binding. But for simplicity purposes, we'll summarize it down to just IP3 binding that leads to the IP3 receptor opening, and then calcium is going to be released from the ER into the cytoplasm. So you generate a calcium signal in the cytoplasm of the cell. Calcium is going to go up. Okay, right, so that's one of the key things that IP3 does. Now let's turn our attention to what the diacyl glycerol does. So diacyl glycerol is really important for activating protein kinase C enzymes. So I'll draw the protein kinase C enzyme here. Okay, diacyl glycerol is one of the utterly key molecules for activating protein kinase C enzymes. The other thing that many protein kinase C enzymes require in order for them to become active is calcium as well. Okay, so often the calcium will work with the diacyl glycerol to activate a protein kinase C enzyme. So this is a PKC enzyme or a protein kinase C enzyme. And then protein kinase C is a serine threonine kinase and can go on and phosphorylate all sorts of different targets and affect activity within the cell, basically. Okay, so phosphoinositide-specific phospholipase C enzymes therefore have crucial signaling roles. They break down this phosphoinositide phospholipid, phosphatidylinositol 4,5-bisphosphate, releasing diacylglycerol and IP3. IP3 causes the release of calcium from the endoplasmic reticulum, and that calcium and the diacylglycerol are capable together of activating protein kinase C enzymes. And the calcium signal can also do many other things in the cell, and then the protein kinase C enzyme can go on and phosphorylate many different targets and have many different downstream effects. Okay, right, so now what I want to mention is that phosphoinositide-specific phospholipase C enzymes, phosphatidylinositol 4,5-bisphosphate, is their absolutely favourite target. That's the thing that they absolutely love to cut up. Okay, however, they can cut up other phosphoinositides as well, okay, notably phosphatidylinositol 4-phosphate. Okay, so let me just discuss this. So they can also break down a phospholipid that's called phosphatidylinositol, so PI, and then it's just got a single phosphate group coming off it. It's 4-phosphate. Okay, so this is short for phosphatidylinositol, okay, and then 4-phosphate. And often phosphatidylinositol 4-phosphate will just be abbreviated down to PIP. Okay, so you can again get away with dropping the PI4P and just abbreviating this down to PIP. Again, if people see just PIP, they'll assume that it means the phosphate group is coming off the 4th one, because that's the most normal one for it to be coming off. Okay, so this is basically just like our phosphatidylinositol 4,5-bisphosphate, but without that phosphate group there. Okay, and again, the phosphoinositide-specific phospholipase Cs, they don't like breaking down PI4P as much as they do PI45P2, but they can do it. Okay, and again, they'll make the same cleavage here, generating a diacylglycerol molecule and then an inositol molecule with just 2 phosphate groups on, the 1st and the 4th here. Okay, so inositol 1,4-bisphosphate rather than inositol 1,4,5-trisphosphate. Okay, finally, phosphoinositide-specific phospholipase C enzymes can break down just phosphatidylinositol itself. So forget these 2 phosphate groups here. You've just got the phosphatidic acid molecule with the inositol molecule. That can also be broken down by phosphoinositide-specific phospholipase Cs. Okay, and the cleavage point will be exactly the same here, generating again a diacylglycerol molecule, and this time just an inositol ring with a single phosphate group off it, inositol 1-phosphate. Okay, right, so that's what phosphoinositide-specific phospholipase C enzymes do, and those are the subjects for this entire video. Right, okay, so what we now want to turn our attention to is these enzymes in more detail, basically. We want to look at the 13 different genes for phosphoinositide-specific phospholipase C enzymes. And we also want to look at this catalytic domain that is common to all of them and how we are going to activate that catalytic domain. Okay, so let's start by discussing the 13 different genes for phosphoinositide-specific phospholipase C. So 13 genes. Now, they are grouped into 6 families, okay? So let me talk about the 6 families before I talk about the specific genes. So, the first family is known as the phospholipase C beta family, and as you can see, we've dropped the phosphoinositide-specific now. Okay, we assume that when we see PLC, that means phosphoinositide-specific phospholipase C. Okay, so when you just see PLC on its own, assume that that is a phosphoinositide-specific phospholipase C. So the PLC beta family. Okay, then there is the phospholipase C gamma family. Okay, then there is the phospholipase C delta family, and I should mention there is no phospholipase C alpha family in humans. Okay, then there is the phospholipase C epsilon family. Okay, Then we get to the slightly fancier Greek letters. There is the phospholipase C zeta family. Okay, so this is the Greek letter zeta. So I'll just write out its name. So if you want to check this symbol, it's called zeta in the Greek alphabet. Okay, then you have the phospholipase C eta family, which looks like this. Okay, so this is eta, the Greek letter eta. Okay, right, and those are the 6 families of phospholipase C enzymes. Okay, now let's go over the members in each of these families. So we'll start with the phospholipase C beta family. So the phospholipase C beta family is the most famous family, and it's also the biggest family. It contains 4 genes. Okay, and these are called phospholipase C beta 1, phospholipase C beta 2, phospholipase C beta 3, and phospholipase C beta 4. Now, these are the ones which are activated by the G-alpha Q subunit of heterotrimeric G proteins, and therefore they are the ones that people are most familiar with generally. Okay, so usually when you see PLC, phospholipase C, in those G protein cascade pathways, usually it's referring to the phospholipase C beta family enzymes. Okay, right. Next up, phospholipase C gamma enzymes. Okay, there are 2 members here. These are phospholipase C gamma 1 and phospholipase C gamma 2. Okay, and these are the ones which are activated downstream of receptor tyrosine kinases, as we'll see later on. That's what they're famous for, although there is another mechanism by which you can activate phospholipase C gamma 2, which we'll discuss as well. Phospholipase C deltas, there are 3 of these. Okay, there is phospholipase C delta 1, then there is no phospholipase C delta 2 in humans. There is phospholipase C delta 3, and there is phospholipase C delta 4. Okay, and these ones have a role in just potentiating the signals of the others, as we'll see later on. There is only one phospholipase C epsilon, which is just called phospholipase C epsilon, and it's the one with the most interesting signaling pathways. It's got signaling pathways that you might not have ever heard of before. Okay, so for instance, it can be activated by RAS monomeric G proteins. It then has a role in positive feedback on the RAS monomeric G proteins, as we'll see later on. It also can be activated by Rho family monomeric G proteins, and Rho family monomeric G proteins are activated downstream of G-alpha 12 and G-alpha 13 alpha subunits of heterotrimeric G proteins, which are activated by G protein coupled receptors such as the thrombin receptors, or the protease activated receptors as they're also called. Okay, and we'll talk more about those later on. Okay, phospholipase C zeta, again there's only one member of this family, and that's just called phospholipase C zeta. This is believed to be extremely important in fertilization, and we'll look at where it's believed to be important later on. Okay, it's believed to be extremely important in triggering the calcium waves that you get propagating across the oocyte when the sperm fertilizes it. Okay, and it's those calcium waves which are really important in triggering the cortical reaction within the oocyte, which prevents polyspermy. So phospholipase C zeta, it may only be found in one little type of cell, which is the sperm cells, okay, but it's really, really important in preventing polyspermy. Okay, right. Then the phospholipase C eta family, this is the one that very little is known about. Okay, there are 2 members here, phospholipase C eta 1 and phospholipase C eta 2, and we think they can be activated by the beta gamma subunits of heterotrimeric G proteins, and we'll come on to those last. Okay, right, so that's the 13 different members, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Okay, right, so now what we want to discuss is principles that will be common to all 13 of these enzymes. So they're all going to be regulated in really different ways. The ways that they are activated are very different. They're regulated by very different proteins. However, there's certain principles that carry through for all of them, okay, and the basic principles are that all of these enzymes are soluble, okay, so they're in the cytoplasm, okay, not attached to the membrane, and if you want to now activate them, you firstly need to get them up to the membrane, you need to attach them to the membrane, and then you also need to activate their catalytic domain, okay, and there's a very special way of doing this, and it's common, we believe, for all first union different phospholipase C enzymes. Okay, so let me discuss the structure of the catalytic domain, because the catalytic domain actually in all of these enzymes is the same, okay, and it's got a really fancy structure, so let's have a look at this. So we're now going to have a look at the catalytic domain of phospholipase Cs. Okay, so the catalytic domain of phospholipase Cs is what's known as a tin barrel-like domain. Whoops, I've missed out the I. Let me start again. Tin barrel-like domain. Okay, so what is a tin barrel domain? Okay, and what's a tin barrel-like domain? Okay, right, so a tin barrel domain can also be called an alpha-beta barrel. Okay, and it's made by alternating alpha helices and beta sheets, as I'll show you in a moment. The reason it's also called a tin barrel is because one of these structures is found in a very important enzyme in the respiratory cascade, okay, which is triose phosphate isomerase, which is really important in glycolysis. Okay, it, remember, turns one triose phosphate molecule into another type of triose phosphate molecule. Okay, I think it, if my memory serves correctly, it turns dihydroxyacetone phosphate into glyceraldehyde-free phosphate, and glyceraldehyde-free phosphate is the one that can then go through the rest of the glycolysis pathway. So triose phosphate isomerase is a very important enzyme in the respiratory cascade. Okay, for short it's abbreviated to TIM, and it has one of these structures in, and hence that's why this structure is called a tin barrel domain. Okay, right, so let me show you what a tin barrel domain is, and basically a tin barrel-like domain is just something that's very similar to a tin barrel domain, but maybe doesn't quite have the exact number of beta sheets and alpha helices that the proper tin barrel domain has. So basically, in a proper tin barrel domain, what you have is 8 beta sheets and 8 alpha helices alternating like so. So let me show this. So here's the amino terminus of the polypeptide, and I'll put a little symbol like this, and what this means is there could be loads of stuff in the protein prior, well, in between here and here. So in between the amino terminus of the protein and the beginning of the tin barrel domain. Okay, so basically I'm just going to firstly draw out the alternating beta sheets and alpha helices, and it will become more apparent when I actually colour these in. So basically, as you go up each time, these are going to be beta sheets, and then as you come down, they're going to be alpha helices. And you're going to have 8 repeats of this, so let me just draw out the 8 repeats, and then I'll colour in the beta strands and the alpha helices in different colours. Okay, right. So again, I'll do this symbol saying there could be loads in between the end of the tin barrel domain and the C-terminus of the protein. Okay, right. So all of these strands going up, these are beta strands, and they're all parallel to one another. So all of these strands in purple, these are beta strands, and they're all going to form a cylindrical beta sheet, basically, as I'll show you in a moment. Okay, and all of the strands coming down, which I'll colour in a different colour, in green maybe, these are going to be alpha helices. So you have alternating beta strands going up, and then alpha helices on the coming down portion, like so. Okay, and this is going to fold up into the tin barrel domain. Okay, so it's going to fold up into one barrel basically. Okay, so let me show this here. Okay, so you're going to have the beta sheets forming a cylinder, or the outer portion of a cylinder on the inside, and then the alpha helices will be around the outside basically. Okay, so here is our tin barrel like so. And basically on the inside you're going to have the 8 beta sheets. So let me just show these in purple. So here's 1, here's 2, here's 3, here's 4, here's 5, here's 6, here's 7, here's 8. Okay, and then around the outside you'll then have the alpha helices. So 1 here, 2 here, 3 here, 4 here, then I might actually show this one going all the way down. Then we've got another one here going all the way down, that we can actually see it going all the way down, and like so. Okay, right, so that's the structure of this basically, that you end up with the beta sheets lining the inside of this cylinder, then the alpha helices on the outside, then a beta sheet on the inside, then the alpha helix on the outside, beta sheet on the inside, and the beta sheets are the beta strands, I should say, rather than beta sheet. The beta strands are forming this beta sheet, which is folded around into a cylinder like so, and that makes the inner aspect of this barrel basically, and that's why it's called a tin barrel domain, or an alpha-beta barrel, because it's made up out of these alternating beta strands and alpha helices. Okay, right, so all of the phospholipase C enzymes are going to contain a structure like this, which is made up of alternating beta strands and alpha helices. Now, the reason it's only called a tin barrel-like domain, okay, is that they don't have the exact same number of beta strands and alpha helices as the tin barrel domain in triose phosphate isomerase, i.e. they don't have the perfect number of 8 beta strands and 8 alpha helices, okay, but the basic principle of having this cylinder of beta strands lining the inside and a ring of alpha helices lining the outside, that's what they do have, okay, so they form this barrel-like domain, okay, which has the catalytic site, which is going to break down the phosphatidylinositol 4,5-bisphosphate inside the barrel, basically. Okay, so let me discuss what the phospholipase C enzymes have in a little bit more detail. Okay, so in all the phospholipase C enzymes, you're going to have a region called the X box and the Y box. Okay, so this is the N-terminus of our phospholipase C enzyme, and again I'm putting that symbol in to say that there could be loads more there, and indeed when we go through each individual example of the, when we go through each of the individual families, we'll see what is in that gap basically. But all of the phospholipase C enzymes have an X box area. Okay, then they have a linker between the X box and what's known as the Y box of the phospholipase C enzyme here, and then they have the rest of this, a gap there, and then the C-terminus. Okay, so this portion in the middle here, this is what's known as the X-Y linker. Okay, so let's colour in these different portions. So here is the X box, which we'll have in green here. We'll have the XY linker shown in vivid purple. Okay, and we'll have the Y box in orange here. Okay, so let me now show you how this actually forms a tin barrel-like domain. So basically this X box region, which I've just coloured in as a blob here, this actually contains alternating beta strands and alpha helices, as does the Y box. And basically these contain the two halves of the tin barrel-like domain that's going to be formed in all of these phospholipase C enzymes. So let me now show this folded up basically. So what we're going to have is this tin barrel-like domain here, okay, and it's now going to be made out of two separate halves. One of these halves will be the X box, okay, so this half here is going to be made out of the X box, so the alternating beta strands and alpha helices that are in this region called the X box, and then the other half of the tin barrel-like domain in orange here is going to be made up by the beta strands and alpha helices of this Y box region. Okay, so here comes the polypeptide off the end of the Y box here, and here comes the polypeptide prior to the beginning of the X box here. Okay, and now what's crucial is that this little site here in the middle of the tin barrel-like domain here, this is where the phosphatidylinositol 4,5-bisphosphate is going to insert its head. Okay, so if I just draw a quick picture of phosphatidylinositol 4,5-bisphosphate, here is the phosphatidic acid molecule, here is our inositol ring, and here are our phosphate groups coming off the 4th and the 5th carbons there. Okay, basically the head of phosphatidylinositol 4,5-bisphosphate, which is the IP3 molecule, is going to get stuck into this cavity here, which is the active site of the enzyme, and then it's going to get its head cut off. Basically, it's going to get this bond between the third alcohol group of the glycerol molecule and the phosphate group cleaved there when you stick your head in there. Okay, so that's the catalytic site of the enzyme. Now, importantly, usually the way that the catalytic site of the tin barrel-like domain is blocked is through the XY linker. So this portion in between the X box and the Y box, it folds over and inserts into that catalytic domain there. So here is this XY linker, this linker between the X box and the Y box, and it usually folds down into the catalytic site, obstructing it basically, so that this tin barrel-like domain is completely inhibited and it can't actually get any PIP2 molecules inserting into it, and therefore it's not capable of actually catalyzing its breakdown reaction. Okay, so this is common to all of the phospholipase C enzymes, all 13 of them. Okay, now let me just discuss then the common features of activation of phospholipase C enzymes. So I've discussed that usually the enzyme is soluble, it's floating around in the cytoplasm. Okay, so if you now want to activate your phospholipase C enzyme and get it to break down PI45P2 into diacylglycerol and IP3, you firstly need to recruit it to the plasma membrane. Okay, so you need recruitment to the plasma membrane. Okay, so that's one of the first steps that you need to do. You need to bring the whole enzyme up to the plasma membrane, and then somehow you need to activate it. You need to remove this XY linker here, and I'll just label this up. This is the XY linker. You need to remove this XY linker from that catalytic site so that the catalytic site can then now actually allow PIP2 molecules into it, and therefore can hydrolyze the PIP2 down to diacylglycerol and IP3. Okay, so the other thing you have to do is get this XY linker out. Now, how do you get the XY linker out? Well, you're helped here, okay, because the XY linker turns out to be negatively charged. Okay, so negative charge. Now, why does that help you? Well, basically, if you imagine the phospholipid bilayer here, let's say we brought the whole enzyme up to the underside of the phospholipid bilayer, so we've recruited it to the plasma membrane. Now, if we want to remove that XY linker, all we need to do is actually repel it in some way, and we can do this with other negatively charged things. So if we bring it in close proximity to negatively charged things, it will get repelled. Now, what is negatively charged? Well, often the heads of phosphate, often phosphate groups are negatively charged, and therefore the heads of phospholipids are negatively charged. So the underside of the phospholipid bilayer is generally negatively charged. So the idea is that what you then have to do is, once you've brought it up to the membrane, you need to reorient it, okay, so that the XY tin barrel-like domain here is oriented so that it's right underneath the phospholipid bilayer. So previously, you might have attached the enzyme up by attaching portions of the enzyme that are in these portions that I haven't shown you yet. And this tin-like barrel domain might be sort of dangling down here. Okay, what you need to do is then reorient it so that it's right underneath the phospholipid bilayer, as I've shown you here. And then what will happen is the XY linker will be repelled by the phospholipid bilayer and therefore gets out of the catalytic site. Okay, so basically the two key steps in activating this enzyme are bringing it up to the membrane, which is usually done by binding to portions that are in these areas, which I haven't shown yet, and which are different for the different phospholipase C enzymes. Okay, and then this tin-like barrel domain will be sort of dangling in the cytoplasm, and then what you have to do if you want to activate the enzyme is reorient the tin barrel-like domain so that it's right underneath the inner leaflet of the phospholipid bilayer. That causes the electrostatic repulsion between the phospholipids in the inner leaflet of the phospholipid bilayer and the XY linker, which causes the XY linker to fall out of, basically, or be pushed out of the catalytic site here, and now the catalytic site is available for PIP2 molecules in the inner leaflet of the phospholipid bilayer to insert into, and then they'll be broken down into diacylglycerol and IP3. Okay, and then once the enzyme has catalyzed one reaction, it will release the products, and then it can do the whole reaction again as long as it's still in this active conformation where it's held right underneath the phospholipid bilayer. Okay, so it can amplify, basically. You activate one enzyme and it will catalyze loads of these reactions, producing loads of diacylglycerol and IP3. Okay, right, so those are the two key steps. Firstly, recruitment to the plasma membrane, and then reorient it so that you flip the XY linker out of the catalytic site here, okay, and then it can now work on PIP2 molecules. Now, this isn't going to quite be true in all examples, okay. In some examples, the way that you get the XY linker out isn't quite just reorienting it to the plasma membrane so that the phospholipid bilayers can electrostatically repel it out. Okay, and for instance, in the example of the phospholipase C gammas, we're going to see that it's actually done by a different means. But the basic principle is that you need to recruit it to the plasma membrane and get the XY linker out, and often that is done merely by reorienting the tin barrel-like domain so that it can be electrostatically repelled out of the catalytic site, basically. Okay, right. So we'll continue this discussion in the next video where we'll start our discussion specifically of the phospholipase C beta family enzymes.